Biotechnology is the integration of natural sciences and engineering sciences in order to achieve the applications of organisms, cells, parts thereof, and molecular analog for products and services. This system uses biological systems. Before dealing with the applications process, we must study the principles of biotechnology. In this video, we'll make a clear picture of the principles of biotechnology, the two main core techniques that enables the invention of modern biotechnology are genetic engineering and bioprocess engineering. How will you define these terms? Genetic engineering is the process in which genetic material is altered in order to introduce into a host organism, thus changing its phenotype or the visual characteristics of the organism. And bioprocess engineering deals with the maintenance of a sterile environment in engineering process to enable the growth of only desired microbe or cell in large quantities for obtaining biotechnological products. Can you guess any such biotechnological products that is in use today? Think about it. Now let us try to learn the conceptual development of the principles of genetic engineering. Do you know the advantage of sexual reproduction over asexual reproduction? Sexual reproduction permits new gene combinations or variations, but asexual reproduction preserves genetic information. So, what is the advantage? New gene combination helps in better adaptability. Traditional hybridization often leads to the multiplication of undesirable genes too. But this limitation has been overcome due to the technique of genetic engineering, such as the creation of recombinant DNA, gene cloning, and gene transfer, which allows us to isolate only desired gene or desired set of genes. Do you know the fate of a piece of DNA that is transferred into a foreign organism? These DNA pieces can't multiply in the progeny cell of the host organism. You may think why it is then transferred to the host organism. These DNA pieces, when integrated itself into the host organism's genetic material, it may multiply and be integrated along with host DNA. In a chromosome, there is a specific DNA sequence known as the origin of replication or ORI region. And this ORI is responsible for initiating replication. So, it is mandatory that for an alien DNA to multiply, it must be integrated into the genome of the host organism. This can be called as cloning. Cloning means the formation of multiple copies of the same fragment. Once the DNA fragments get incorporated into the host DNA, we call it as recombinant DNA. How will you construct an artificial recombinant DNA molecule? This idea has first emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid of Salmonella typhimurium. What is a plasmid? Can you define plasmid? Recall your class of grade 11th where it says that plasmid is the circular autonomously replicating extra chromosomal DNA present in a bacterial cell. Stanley Cohen and Herbert Boyer accomplished this in the year 1972 by isolating antibiotic-resistant gene by cutting a piece from the plasmid DNA, which ultimately confers antibiotic resistance. How are the insertion in DNA done? The cutting is done by restriction enzyme. It is also called molecular scissors, which are meant to cleave DNA at specific sequences. The cut piece of DNA is linked with the plasmid DNA and they act as a vector. They transfer the alien DNA into the target organism. Have you ever thought how the malarial parasite reach our body? Mosquitoes transfer malarial parasite into the human body. Hence, Mosquito here act as a vector. We have seen that a DNA fragment is incorporated into the host. What helps in the ligation of these fragments? Is there any enzyme to do so? What is the role played by DNA ligase? DNA ligase is the enzyme that links the alien DNA with the plasmid that acts on the cut end and joins them. This makes an in vitro copy of autonomously replicating DNA or the recombinant DNA. If it is transferred into E. coli similar to Salmonella typhimurium, DNA polymerase makes multiple copies of the recombinant DNA. Finally, there are 3 steps in genetically modifying an organism, and they are identification of the DNA with desirable genes, introduction of the identified DNA into the host, and maintenance of introduced DNA in the host and the transfer of the DNA into the progeny. Biotechnology is an important and very vast topic. In this video, we have only discussed the principles and core techniques. It was mentioned that the restriction enzyme helps to cleave the DNA at specific sites. How are the specific sites decided? Try to find it out. Let's summarize what we have learned.
Manorama Horizon. Learning made simple.